Hey everyone, thank you all for being here for the third and Arkansas Native Plant Society's 2024 webinar series. We're very excited to have Benton with us here today as for his talk titled Untangling the Biotic and Abiotic Factors that Structure Prairie Pimple Mound Plant Communities. Benton is a botanist for the Arkansas Natural Heritage Commission and the curator of the Natural Heritage Commission's Herbarium. Ben graduated from Hendrix College in 2020 with a degree in biology and is currently pursuing his master's degree in biology from Arkansas State University. Ben is interested in the ecology of diverse plant communities, and his research at A-State investigates the role of soil nutrients and microbial communities associated with patterns of diversity in Arkansas's remnant prairies. It's pretty exciting. The third and the fourth one webinar in this series, the other one coming up later this month, both have a soil component, which I think is it's going to be really interesting to hear. So we're really glad to have Ben here to talk to us about prairie pimple mounds. A recording of today's program will be uploaded to our YouTube channel, and I encourage you to go there and view all of our other webinars that we've hosted over the years as well. If you'd like to learn more about the Arkansas Native Plant Society, you can do so by visiting our website at anps.org. You can join the Native Plant Society right there on our website. You can even do it today from your home or computer or device. Just go to anps.org/join, and you can use your PayPal account to join today. So if you would like to, if you don't have PayPal or prefer not to set up an account, you can send a check to our treasurer, and their name and address should be right there on our website. And so I'll drop some links in the chat here for everyone. But without further ado, I'm going to hand it over to Ben. Thank you, Ben.
All right, thank you. Yeah, so thank you for the introduction. That's pretty good. So yeah, this presentation is over the research that I did while I was at Arkansas State for my thesis work. So just kind of jumping right into it, kind of answering the question of what exactly is a community in an ecological sense. Basically, a community is the species that occupy a given space or occupy a given space at a particular time. And so generally speaking, you have kind of this ecological hierarchy where you have individuals that are of a particular species. And then whenever you have multiple individuals, that forms a population. And then whenever you have populations of different species, that forms a community in a given area. And these community or these populations will change in, or the communities will change in what species are present and how abundant they are. Their relative abundance within the community will change over environmental gradients and respond to a variety of factors. Just kind of a brief kind of synopsis of how communities have been studied in the past. So a common kind of metaphor for a community is a jar of candy, where each individual type of candy represents a particular species, and then all of the candy of that particular type within a jar represents a population, and then the whole candy jar itself is a community. And so here I have kind of three examples of a community of candy in a jar, with the composition of each jar is different, and the abundance of each candy within the jar, it differs from community to community as well. And so kind of for this little exercise, we'll say that there's environmental differences between where these candy jars, whose desk these candy jars are sitting on. So these two candy jars, can you see my mouse? Hopefully you can see my mouse. So like the two candy jars on the left.
Yes, we can see your cursor.
Okay, good. So the two candy jars over here might be like in the basement where there's no windows or something like that. And this candy jar right here on the right is sitting on the boss's desk in the big office with the window. And so there's kind of differences in the environment that might have an effect on what's there. So the way that we can compare communities is by looking at calculating their similarity or perhaps how dissimilar they are. And so there's dozens of different metrics that can be used to calculate this, but one that is pretty frequently used and one that I use in my research is the Bray-Curtis dissimilarity index. So it's a measure of how different things are when I'm saying dissimilarity. So if you have two, and it takes into account the species that are present in the community as well as how abundant those species are. So if you have, it will capture differences in if you have identical species in a community, but the one species is like super abundant in one community and the same species in the other community is super rare, it will capture the dissimilarity there as well. So if you have two candy jars that are completely identical, you'll have a dissimilarity of 0. But if you have candy jars that are, or communities that are completely different from each other, they have total turnover in the species that are there, then you'll have a dissimilarity of 1. And so you can compare these dissimilarities with each other. So these are the Bray-Curtis dissimilarity values calculated for each of these candy jars and how similar the composition of these candy jars are to each other. And you can compare these to the environmental factors of where these candy jars are, and you can kind of draw conclusions based on relating dissimilarity to some environmental factor. And you can start to kind of infer how these environmental factors might affect the community composition. So in this, I think my thought process was the chocolates in the candy jar melted whenever they sat in the window. So you kind of see a turnover. You see that replacement of the chocolate with the hard candy in this exercise, and that's that. So you can see an association of the community composition with how much light is in the environment, so to speak. So this process is called community assembly. And so kind of the modern concept of community assembly is that species from a regional species pool, so just species in the area will migrate to a particular point, and if there's adequate resources there, then they will be able to establish. And then there are abiotic filters in the environment. So whether or not there's enough like resources in the environment for that species to establish, and the particular levels of those resources will affect how abundant a particular species is in a community. And then there are biotic filters, which are things like interactions with other living organisms. So you could have predation or pathogen effects from microbial interactions or beneficial mutualistic interactions with living things, all kind of act as a filter that will that determine the relative abundance of a particular species in a community and their ability to persist within the community. So there's been a lot of research in the past that has kind of focused on these biotic and abiotic filters and describing how they function, and they're really well supported. But there's not been a ton of research that has compared how these two filters interact with each other to influence the relative importance of each other. And so that's kind of where the niche that my research kind of fits in there. Is I'm kind of trying to address the question is, how do these changes in biotic and abiotic factors in the environment relate to changes in the community, and in my particular case, in the plant community. So to do that, my study area, I chose Cherokee Prairie Natural Area because it's one of the largest tracts of Arkansas River Valley tallgrass prairie at 584 acres in Franklin County. If you guys have never been out there, it's super beautiful in the spring and summertime. But it's a super diverse community, which is one of the reasons that I chose it. So it has a history of being the flora of it being documented all the way back to like the 70s, and there's 345 species of plants known from this site. And I also chose Cherokee Prairie because it has these really cool things called pimple mounds that are there. So pimple mounds have very visually distinct plant communities from the matrix that they kind of exist in. So the intervening prairie matrix, the plant communities are very visually distinct. And there's also a known abiotic gradient from the mounds into the prairie matrix. So typically, when I talk to people about my research, their first question that they have is, what the heck is a pimple mound? So essentially, a pimple mound is just this small hill. They're typically found in prairies or associated with prairies in some ways, but they're about 1 meter tall and 20 meters in diameter, just a little hump out in a field, kind of. And they typically have sandier soil than their surrounding prairie matrix that they're in. And they're kind of particularly sought after by conservationists because they're associated with, they're associated with high quality ecosystems and really intact ecosystems. So if we look at the LiDAR here, one kind of particular thing that conservationists will clue in on when they're looking for, like, a piece of land to protect is how intact the pimple mounds are. So they're kind of delicate features. So you can kind of see across this fence line where the prairie has been plowed. You can kind of see the ghosts of pimple mounds past. But if you have an area that has really nice, robust, intact pimple mounds, you can kind of infer the history of the land use there and say, like, oh, well, I can tell that this area has never been plowed, and the ecosystem is probably pretty intact. So they're pretty useful for conservation efforts. But they have a really interesting origin. The first use of the name pimple mound that I can find reference to is this 1962 master's thesis called "The Origin of the Sand Mounds of the Pimpled Plains of Louisiana and Texas." And there's been a lot of hypothesis kind of where these mounds came from. So things like burrowing rodents and swelling soil deposits or deposition of the soil from ice or wind or water have all been published as possible hypotheses for where these mounds have came from. But the theory that I put the most stock in and think is probably the accurate theory is from the Seifert et al. Paper, which is a really, really cool paper. That suggests that these pimple mounds are actually these relict sand dunes called nebkhas. And nebkhas are formed today, and they're essentially a sand dune that forms from soil being blown in the wind and catching on like a shrub or a clump of grass or something like that, and it forms a dune. But the evidence that supports this is that this paper described this gradient of soil texture across these mounds. So the soil was coarser on one side and more fine on the other side, and it was all directionally a directional gradient, and it changed from or it was the same from mound to mound across the site, and it was very similar from site to site across the state. So and they were able to estimate when these mounds formed, and it kind of lined up with this drought in the late Holocene era, about 5,000 to 2,000 years ago, if I recall correctly, that was kind of sent Arkansas into these like dust bowl like conditions where these sand dunes would have formed, and kind of there was severe desertification in the state. And so then when those drought conditions ended, plants grew back across these mounds, and it stabilized the dunes, and they're still around today.
Ben,
Yes.
we have a question in the chat. Are pimple mounds the same thing as Mima mounds?
Perfect. So this goes into my next slide here. So yeah, so this has been very hotly debated. That was a perfectly timed question. This has been something that's been so debated that there's even publications that that kind of call into question, or that consider it a controversy of why there's so many theories of where these mounds came from. And so I think that is because there are a ton of different mounds or mound-like features in grasslands across the country. So this is a publication from that is talking about these mounds, but particularly Mima mounds, like you mentioned. And so you can see that across the country there are mounds in prairies are super commonplace. And so Mima mounds is a common name for calling for pimple mounds. They often get called Mima mounds, and that's kind of the current paradigm, is that all of these mounds across the country and across the world too. There's mounds, prairie mounds in South Africa and in South America and Australia as well that I've seen or seen documentation of. But the current paradigm is that all of these get called Mima mounds. And I think that kind of contributes to why the history of these mounds is so controversial, is because everybody's describing these unique origins for their particular local mounds that they're studying. And but really they all have different origins, but they're all getting called Mima mounds, which have, their own particular origin. So a Mima mound is I think specifically, I'm making the argument that a Mima mound specifically refers to a mound in these Puget lowland prairies in Oregon and Washington. This is the type locality of where the term Mima mound came from. This is Mima mound, an aerial photo right here of Mima Mound Natural Area Preserve in Washington. And this is me standing next to a Mima mound there. So I got to visit the site because I'm a mound enthusiast. But that same publication that mapped the mounds makes the case that that Mima mounds should refer to only mounds in the Puget Sound or Puget Prairie area. And he says that all other mounds should be referred to as Mima-like mounds to kind of make that distinction. But I think that because we have a history of referring to these mounds as pimple mounds, I think that we should specify and call these mounds pimple mounds to kind of be more specific about their particular origin and their ecology that I'm about to get into. So I say, most people say that pimple mounds are Mima mounds, but I say that they're different. That's kind of my case. So just so we are totally clear, pimple mounds are small hills that commonly occur in prairies in and around the vicinity of Little Rock, and Mima mounds are small hills in prairies that occur in the vicinity of Little Rock. Okay, so we're crystal clear on that. So getting into the ecology of it. So the first step in studying, or in my study, was to describe the spatial variation that we saw at Cherokee Prairie. So the differences in the plant communities, as well as the biotic and abiotic variation at or associated with the mounds and the prairie matrix at Cherokee Prairie. So the way that I did that is I described or took data on the soil nutrients, the plant communities, and the fungal communities across 20 sampling sites, where I had two paired transects, one that went across the center of the mound right here, and one that was 25 meters off to the east or the west of the mound here. And so I did that across 20 sites. This is a map of my sites. And each one of these dots represents a 1 meter plot that I collected data on the plant communities that were there. So me and my lab mates recorded the plant species that were there and their relative abundance. And we also collected soil samples from each of these plots and used the MELIC3 extractant technique to kind of quantify the soil nutrient profile of each of these plots. And then I also collected the extracted DNA from the soil, from the microbial communities of the soil, and I looked at the fungal communities using a metabarcoding technique to kind of describe the fungal communities that were in the soil as well, associated with the plant communities. So here's kind of a visualization of the results of that. So overall, I found 100 species in my study. 73 of these species were, or I found 73 species on the mounds and 53 species in the matrix. And 47 of these species were exclusively found on mounds, and 27 of these species were exclusively found in the prairie matrix. So this is kind of a visualization. So thinking back to that dissimilarity with those candy jars, calculating the dissimilarity of those, this is a visualization of the dissimilarity of the plant communities at Cherokee Prairie. So basically the way to interpret this, this is an ordination plot of the community there. And so the way to interpret this is each one of these dots represents a single plot from my study. And the closer any of these two dots are, the more similar the plant communities are to each other in the species that are present and the relative abundance of those species. So you can see that we have really distinct differences in the plant communities between the Pimple mounds and the prairie matrix. So just to kind of drive this home, you have, you see the differences in these, or the prairie matrix communities are more similar to each other than they are to Pimple mound communities and vice versa. So this particular plot right here, the plant community there is more similar to the plant community in this plot way over here, because they're both in the matrix, than this plot is to the Pimple mound community that's just 25 meters away from it. So we see this distinct turnover in the plant communities at Cherokee Prairie. And what's really cool is we see that exact same pattern in the in the fungal communities and the soil nutrients. So moving from the Pimple mound into the matrix, we see a great dissimilarity in the plant communities, fungal communities, and soil nutrients. This is kind of the stats of that. So yeah, so basically the mounds had different plant and fungal communities than the matrix did. They were more similar to other mound communities than they were to matrix communities. And the mound soil, or the soil, was also followed that same patterns. And mound soil typically had higher levels of nutrients than the matrix soil did. One particular kind of thing to note that was interesting is the mound soil had 6 times the amount of phosphorus than the matrix soil did. So the mounds are just this super nutrient rich soil compared to the matrix soil. And so this is the results of a partial Mantell test. So this is a statistical test that looks for correlation in dissimilarity. So I'll break this down for you pretty simply, I think. So the way that, or whenever you look at the all of the communities and all of the soil nutrients across the site, this test found that the more similar any two communities were, any two plant communities were, the more similar the fungal communities were. And the more similar the plant communities were, the more similar the soil nutrients was. But if you break that down and look at just the mounds, we kind of see this interesting shift. So on the mounds, we found that the plant communities were, the more similar any two plant communities were to each other, the more similar the soil nutrients was. There was a relationship between plant communities and soil nutrients on the mounds. But there was no relationship between plant communities, how similar two plant communities were, and how similar the fungal communities were. But whenever you go 25 meters off the mound and you're in the prairie matrix, you see a switch. So the plant communities were more associated with, or were associated with the fungal community similarity. So the more similar any two plant communities in the matrix were, the more similar the fungal communities were. But there was no relationship between the plant communities in the matrix and similarity with the soil nutrients. So moving off the mound into the matrix, we kind of see this shift from plant communities being associated with soil nutrients to the plant communities being associated with the fungal communities. And across both the mound and the matrix sites, we see that the more similar the soil nutrients are, the more similar the fungal communities are. There's a correlation between the fungal community similarity and the soil nutrient similarity. So that's a little confusing, but basically they swap from the plants being associated with soil nutrients on the mound to fungal communities in the matrix.
Ben, we have
Yes.
another question in the chat, and I'm not. Val asked, great variation in pimple mounds too. Maybe Val can clarify what she's asking or what they're asking.
Yeah, so I think I kind of understand that question.
Okay.
So yeah, there was, and you can see it in this NMDS plot right here, that that generally speaking, the plant communities in the matrix were more homogeneous. So the plant communities were very, very similar to each other in in the prairie matrix, and that's why we have this kind of tight clustering of of points right here, but there was more variation in the species present and species abundance in the pimple mounds. And so it kind of forms this larger cloud these mound plots do. So I think that kind of answers that question. There is more variation in the plant communities on the pimple mounds.
And I have a question. Did your study at all look at like soil moisture, if there's a correlation there? I know here in Northwest Arkansas, between the prairie mounds, we tend to get like extra soil moisture, and sometimes you can even have more mesic communities or a lot of like sedges and whatnot. So I didn't know if looking at the plant communities that were, but not on the mounds, did they seem to be? Maybe you'll go into a little bit of what species are most common, but just wanted to ask that.
Um, so that's not something that I particularly address in the field component uh of my research, is like how the soil moisture affects or relationships with um the the plant communities to kind of the physical properties of the soil. All of it was kind of looking at the soil nutrients, because we're kind of exploring, or I was kind of exploring the trying to kind of quantify that interaction of the microbes with the soil nutrients, because there's that important relationship of fungi. They can either be pathogenic or mutualistic in helping fungi kind of more, or helping the plants more efficiently uptake nutrients. So I didn't really look at the physical properties of the soil necessarily in the field study, but kind of just moving on to the greenhouse experiment, that does kind of, I do kind of address that somewhat.
And I'm sorry, we have another question in the chat. Josh
Yeah.
Poland asks, was there any variation in diversity with different heights of the pimple mounds?
Um, that is not, hi Josh, by the way. That's not exactly something that I explored in this, um, but the way that the plots were set up, they kind of were, let's go back to, so we kind of had two kind of lower plots and a higher plot that was located in the center of the mound, and then two that were kind of off the edges of the mound on the north and south side. And those would have been kind of lower. But we didn't really see a difference in the plant communities kind of associated with the topology of the mound. And the mounds are all pretty uniform height from like mound to mound. And so there may be a difference between, these like really minute changes in the mound height, but that's not something I've explored yet. So I'm not sure if there's a relationship with that kind of micro topography, so to speak, and the plant communities. Let me jump forward again. Yeah, so the field component of my research found this really clear and interesting pattern in spatial variation at Cherokee Prairie in all three, in the plant communities, in the fungal communities, and in the soil nutrients. But it doesn't really address the question of how these environmental qualities are interacting with each other to affect the plant communities. And so to answer that question of how these are interacting with each other, we needed to do a greenhouse experiment. So I started out by collecting seeds from 8 different species at Cherokee Prairie. I collected seed box, slender mountain mint, prairie blazing star, Missouri ironweed, southeastern wild rye, Indian grass, broom sedge, blue stem, and big blue stem. And those were just plants that occurred at Cherokee Prairie that had a seed abundant enough for me to collect for my experiment. And then I also collected soil from either the prairie matrix or a pimple mound from property that was just adjacent to Cherokee Prairie. It's a sensitive site, and I didn't want to disturb it by collecting a bunch of soil. So we just hopped across the property line, and it's just a field that it's been mowed for hay. And so we collected soil from there to kind of match that, or to kind of capture the differences in the soil characteristics. So we then took that and carefully set up the treatments of my experiment. So there were, with 8 species, we had 6 different soil treatments and 10 replicates. And the way the soil treatments were set up is I took the soil that I collected from either the mound or the matrix and autoclaved it to sterilize it and kind of remove that the abiotic or the biotic factor from the soil. So it was just the sterilized soil with no microbial community in it. And just kind of and use that as the abiotic treatment in my experiment. And so each one of the soil treatments had a plant planted in it. And then I took a small kind of aliquot of soil that had not been autoclaved from both the prairie matrix and the pimple mound, and used that to inoculate each of my sterilized treatments with a small amount of the microbial community associated with each of those kind of habitat types. And so I had each combination of plant, soil type, abiotic soil type, and microbial community for each of my treatments. And then, not pictured here, I also had a treatment of plant in either mound or matrix sterilized soil that did not receive an inoculation, that kind of served to act as a point of comparison for the effect of the microbial community on these plants. So I also had a sterilized treatment. My lab mates helped me assemble that, and I grew that in a controlled greenhouse environment for 71 days in the late summer of 2021. And then after that 71-day period, I harvested all of the above ground material, and then we meticulously washed all of the soil out of the root systems, and collected those as well. And then those were dried and then weighed to collect data on the biomass. So here are the results of the greenhouse experiment. So we found that in general, plants grew considerably larger whenever they were planted in mound soil. And we did not find, between both the mound and the matrix treatments, there was no effect of the source of the inoculum. So both microbial communities from the mound and the matrix performed about the same. We did see that that plants that were planted in matrix soil there was a very large effect between the of the biomass of the plants associated with whether or not there was actual microbes present in the soil. So in the matrix soil, we found that inoculated plants grew considerably bigger than plants that were growing in just sterilized soil. And that's not an effect that we saw in the mound soil. So sterilized plants performed just as well as plants that were growing in with a microbiome or with a microbial community in the soil. But we saw that plants growing in sterile soil, sterile matrix soil, basically didn't grow at all. They never developed kind of past a seedling stage. They just kind of were like sitting in stasis, alive but not really developing. And plants that were plants in matrix soil that had microbes kind of developed into like somewhat normally. So what's really interesting about that is it seems like our field study and our greenhouse study are kind of telling different parts of the same story here. So if you think back to the results of the field study, we saw that plant communities at Cherokee Prairie were associated with fungal communities in the matrix and soil nutrients in the mound. And across both of those sites, there was an association with the fungal community and the soil nutrients. And so looking at the results of the greenhouse experiment, that kind of drives home and reinforces this idea of the relative unimportance of soil microbes in mound soil. So we saw no effect of inoculation on plant growth in mound soil, and the importance of soil microbes in the matrix soil. So whenever plants were growing in matrix soil, it really mattered if there were microbes in the soil versus if there weren't. So kind of in conclusion, the results of these two studies kind of agree with each other that there's this asymmetrical effect of these soil microbes on the plant communities. And that effect switches from being more nutrient driven on the mounds and more microbially driven in the prairie matrix. And the results of these suggest that the soil characteristics, the underlying soil characteristics, are what are influencing kind of the strength of the effect of these microbial communities in how these plant communities are assembling. So
And Ben, we have another question in the chat. David
yeah.
Berger asked, how hard was it to identify the fungal species?
So it was both pretty tricky and very easy at the same time. So I did a metabarcoding. It was a metabarcoding study. So all I did was extract the DNA from the soil of everything living in the soil, both fungal, bacterial, and nematodes, and you name it. And so that particular aspect of it was super technical, doing the DNA extractions. And then but the actual identification process was relatively easy because we just sent the samples to a lab. They sequenced the DNA for us and gave it back to us. And then we compared those DNA sequences that we pulled out of the soil to a reference library of these DNA sequences. And we were able to assign species to each of these DNA sequences that we extracted from the soil. So it was not necessarily like, there are some techniques for identifying like fungal spores to species under a microscope and stuff like that. But the approach that we used was to kind of just use DNA to identify what species were present.
Val asks, so why do you speculate nutrients are so high in the mounds?
Yeah, that is a good question. And um, if I were a better soil scientist, I might be able to have an answer for you. I'm not sure why they are particularly higher. Maybe because it's relatively newer soil and like there hasn't been, you know, this history of nutrients being extracted from it in the from plants growing there. Or my kind of one of my personal hypotheses is that the matrix tends to flood in the wintertime. And so I suspect that burrowing rodents might like to burrow on the mounds versus the matrix where their burrow would be flooded, so they're kind of burrowing in high ground, and so there might be this vector of moving nutrients, like the rodents might be foraging in the matrix and then going and living and like defecating and moving that nutrients from the mounds or from the matrix onto the mounds. But that's a whole different master's thesis, and I'm so, but that's just my hypothesis as to one reason that it might be. But yeah, I don't know, but it's really fascinating. But yeah, so that's kind of it for my presentation. I'd like to give a few thanks to some very awesome people. My lab assistants and my field assistants, and particularly Kat Frank was my undergrad assistant. She was super awesome and very pivotal in collecting data from the greenhouse experiment. She basically single-handedly washed all of the roots from like the 500 plants that I had, wash the soil from the roots. It's a very difficult and meticulous job that she did, and I'm super grateful for that. But Brendan Kosnick, Grace MacArthur, and Katie Sims were also awesome lab mates, super helpful in getting this done. My committee members at Arkansas State, Scott Mangan, Travis Marsico, and Kyle Gustafson are an awesome committee. And I'd like to thank my funding sources as well, the Superb Scholarship, Arkansas Game and Fish, and of course, last but not least, the Arkansas Native Plant Society for providing funding for my research. And then as well, I'd like to thank my friends and family for the support along the way. And yeah, I'll use my remaining time for any questions that you guys have.
Yeah, thank you, Ben. That was amazing presentation, really interesting research, and we really appreciate you sharing it with us. And Native Plant Society, one of the things that we do, of course, as you know, Ben, is funding research and students to do research that benefits or expands our knowledge of the native plant communities in Arkansas. So we were happy to help fund you, and so just letting people know when you join the Native Plant Society, your dues, that's what your money is going towards, is funding research like what Ben is doing here. We have one question in the chat. David has a follow-up question to his earlier question about the fungal species. He asks, how much did it cost to get the DNA analysis of the sample?
Yeah, so um, it cost about $15,000 to do the meta barcoding, to have the in that was equipment. So um, we that was buying kits for DNA extraction. They're pretty accessible and easy to use. Um, but so that cost quite a bit, and then also um, the actual cost of having the DNA amplified and sequenced in a lab was about $15,000. So it is a little expensive. That's why my funding is so nice. Got to thank them.
Yeah, if anyone else has any questions, I'm sure we're at a point where if you would like to unmute your microphone and ask Ben, feel free to go ahead. Last call for questions. think people have probably been asking the questions they had throughout your presentation, but we'll give one last call. All right. Well, again, thank you, Ben, for sharing your research with us. It's been really interesting to listen to. Uh, Really interesting research. I think pimple mound communities are something that there needs to be more awareness of, because there's still a lot of those remnant communities here in Northwest Arkansas, my part of the state, and we see a lot of these areas getting developed and plowed over, and we really are losing those intact ecosystems. There's just not much focus on or awareness over what those ecosystems are. So I want to thank everyone else for being with us here again today. Again, the recording of this webinar will be uploaded to our YouTube channel, so please check out our YouTube channel. Drop the link in the chat. Oh, it looks like we have Greg asked a question, so I'll if you can hang on one second, Ben. Is
Yeah,
there a
so
map of Pimple Mound locations in Arkansas?
yeah, so the closest thing I've seen to a map of these mounds is from this publication right here, which I guess I didn't write down the actual title and author of this that I got this from, so that's a mistake for on my part. But this is a map of prairie mounds across North America, and so it kind of includes these. What I would consider pimple mounds here in kind of Missouri, Oklahoma, Arkansas, Louisiana, and Texas. So that's kind of a pretty general map. And it's the only one I know of. I'm not sure exactly how accurate it is.
Yeah, and it's interesting because as you really learn to identify these as you're riding around, sometimes you'll spot little areas where they're remaining. So I mean, I've run into them doing field work in Northeast Texas, Northwest Louisiana, Western Oklahoma, and even different parts of Central Arkansas. So yeah, it's pretty cool once you realize what you're looking at.
Yeah,
But
you see them everywhere. It's really fun to spot it, and everybody that I hang out with is always rolls their eyes when I'm like, we're driving somewhere. Like, oh, pimple mound. I'm like, that's a pimple mound over there.
Yep, I know. I get excited too. Yeah. We need one of those jars where you put a quarter in every time you identify a pimple mound without being asked. So,
Yeah, exactly.
Adam asks, are the plants on the mounds more likely to be rare species, or are they just different than the matrix locally?
Yeah, so that's a really good question, and also good to see you, Adam. So uh, the that's not exactly something I've done an in-depth kind of like analysis of. But generally, I would consider the species that I see on the mounds to be a little more weedy than the species that I see in the prairie matrix. But kind of an avenue that I'm really interested in exploring now is very recently there was a publication of the coefficients of conservatism values for plant species in Arkansas. So kind of a measure of how conservative plant species are. And so I'm really interested in seeing if there's any association with those levels and the species that are like, if there's any association with how conservative the plants are and whether they grow on the mounds or the matrix. So that would kind of be an investigation into how rare like if there's any association with the species rarity in these two different habitats. So that's kind of that is an interesting like avenue that I'm hoping to explore. So yeah.
And those C values, I believe, are now available on Phytoneuron. Is that correct?
Yes, that's correct. Adam's follow-up question. So the ones that were less nutrient driven were the ones in the matrix. So the nutrient driven communities are on the mounds where I see the weedy species. So, or what I would associate as weedy species, being on the nutrient driven and high nutrient soil on the pimple mounds. And then kind of the more conservative, less weedy species being in the more microbially driven prairie matrix.
In that paper, going back to Greg's question about the maps of the mounds, that paper, the 2009 paper, was it Siegert? Is that the last name? I believe it's
Uh,
on the slide before that one.
yeah, Seifert.
Seifert,
It's
yeah.
Seifert and Tom Fotai is on the paper too. It's a really good paper.
Yeah, and it seems like there was a map in there of Arkansas, and it might have just been the different locations where they had sampled or did their study. I can't recall.
Yeah, so this figure right here is from that paper, and these are just the four different study sites that they did. So um, this right here is Cherokee Prairie, um, where I did this research. So that was actually part of their study, was mounds at Cherokee Prairie. And then this is Warren Prairie down here, and this is a site just outside of Searcy, and I can't remember what this one in the middle was.
Looks like, yeah, river valley of some kind is what I assume that RV
Yeah,
stands
that might
for
be what
there.
that is.
But all right. Any other questions for Ben? So if you're interested in learning more about the interactions between plants and soil, our next webinar on November 16th will be at 5 p.m. That's a Saturday. Justin Thomas is going to give a program on the amazing world of wild plant soil interactions. Oh, it looks like we have another question in the chat. Adam asked, just reminded me that in California, they are seeing non-native weeds and grasses invading serpentine low nutrient soils due to nitrogen deposition by air pollution. Interesting.
That is interesting. And I, I feel like we kind of have an opposite problem in Arkansas, where I'm hearing cases of like where there's contamination of areas with fertilizer, where there's being nutrients added um, to kind of these sensitive areas, that's allowing for like invasion of less conservative species. So uh, here in Arkansas, we're hearing about or like I'm hearing about kind of the opposite effect of that. Um, well, actually, yeah, no, that's the would be the same where it's where yeah, never mind. I misread that. Sorry. Yeah, we're kind of seeing the same problem in Arkansas. But
Sounds like a different source of that
yeah,
nitrogen
different
pollution,
sources.
air pollution versus yeah, fertilizer.
It's interesting. Yeah, these super sensitive habitats associated with kind of low nutrient levels, like is very interesting.
Yeah, and I assume Justin Thomas will be going into that a little bit in his presentation. If you I don't know if you know Justin Ben, but
Yeah.
yeah, that's one thing he talks about, or yeah, the C values and how they relate to soil nutrients and nitrogen and all that sort of stuff. So
Yeah.
should
Yeah.
be an
I definitely
interesting talk.
see the parallel with what Adam's talking about.
And I believe Justin was developed the C values for Missouri, if I'm not mistaken, or wrote that paper. But I could be wrong on that, but I know his name's on a paper that has the C values listed in there. All right. Well, we are at the top of the hour, so I will go ahead and if you have any questions for Ben, I don't know if you want to drop an email in the chat if people
Yeah,
want
I'll
to
put my
follow up
email
with you.
here.
Yeah, that'd be good. And again, if you're interested in learning more about the Native Plant Society or you want to join so that your membership dues could help fund more research like Ben's. And also, you get access to our journal and membership meetings, which we have twice a year, once in spring, once in fall, where we have plant hikes and all that sort of great stuff and get to meet other people that are into this sort of thing. You can go to our website, anps.org/join and PayPal account to join right now today if you would like to. You can follow us on Facebook for more of these webinars and other content, facebook.com/Arkansas Native Plant Society. And again, follow us on YouTube. Just go into YouTube and search Arkansas Native Plant Society, and you should be able to find us there. And everybody, I hope you enjoy your rest of your weekend. Thank you again, Ben, for a fascinating talk. We really appreciate you sharing your research. You're doing great work.
Yeah, thanks for having me.
Thanks, everyone.